Hello students, in this session we will discuss transgenic plant. We will discuss a topic given in NCERT. It is related to transgenic tobacco. In your book they mentioned tobacco can be protected from nematode infection by novel strategy, and this novel strategy is called RNA interference. Okay, so topic is transgenic plant, pest resistant plant. This is NCERT paragraph. In this, they are talking about nematode Meloidogyne incognita. This nematode infect tobacco plant, root of the tobacco plant, and it destroy tobacco plant. Tobacco is a cash crop for farmers. If this crop is destroyed, it can affect income of the farmers. Now, scientists they have created one pest-resistant tobacco by using RNA interference. Okay, name of the technique is RNA interference. So, first of all, you can focus on this paragraph. RNA interference. Who was first to discover RNA interference? RNA interference was first discovered by Craig Mello. Okay, they along with Andrew Fire in 1988, and they also got Nobel Prize for this. Okay, so they discovered RNA interference, which and they have not discovered RNA interference in Meloidogyne incognita. They discovered this RNA interference in one more nematode, and that nematode is called Cenorhabdites elegans. Okay, so this is not given in CRD, but this is background. Okay, and again I'll ask a question: who discovered this RNA interference? Craig Mello and Andrew Fire. In which nematode? Cenorhabdites nematode. Okay, so in RNA interference we will study mechanism, how this RNA interference operate in eukaryotes. Okay, you can assume this is eukaryotic cell. This is eukaryotic cell, and in this eukaryotic cell you can assume this is cytoplasm. This is cytoplasm, and this is nucleus. Okay, this is cytoplasm and nucleus. Already it is discovered by scientists that in eukaryotes there is one mechanism. It is called RNA interference, and in this RNA interference there is one term RISC. What is RISC? It is RNA induced silencing complex. What is RISC? RISC is a basically complex of many proteins plus RNA. Okay, if this type of complex is activated, then this complex it can destroy RNA. You can see these are many proteins, and these proteins are associated with this RNA. If this RNA is attached here, it is activated complex. This type of activated complex exists in eukaryotic organisms. Now this activated complex, if it is attached to complementary RNA, this type of RNA, mRNA, okay, this RNA is having sequence complementary to this RNA. So now these enzymes they can destroy this RNA. So this RNA is converted into fragment. Okay, it is broken into fragment, and this is now it will not participate in translation. Okay, so it will not produce any protein. Okay. Now question is how it is activated. How it is activated? To activate this we need one RNA. Okay. Now source of this RNA can be exogenous RNA coming from external medium. This RNA can activate this. Okay, what is source of this we will study here. Or RNA which can activate this may be RNA of the virus. Suppose this is virus, it is having double-stranded RNA. Now this double-stranded RNA, it may be broken into small segment. It is broken into small segment. This small segment is called siRNA. siRNA is a short sequence. It may be a 21, 22, 23 nucleotide long sequence. This siRNA is produced from viral RNA with the help of enzyme. This enzyme is called dicer. You can see it is an endonuclease. It cut this RNA into small segment. Now this small segment which is double-stranded, this may initiate or this may activate RISC. Now from this double-stranded RNA, one strand bind here and second is removed. Now this one strand which is attached here, it will activate RISC. Now in future, if RNA is trying to multiply inside the cell, it produces multiple RNA of the virus. In future, if RNA of the virus is trying to multiply inside the cell, then this RNA of the virus, suppose this is the RNA of the virus. This is the RNA of the virus. This RNA of the virus can be destroyed by RISC. If RISC is activated and this RNA virus is trying to multiply inside host cell, then this RNA virus may be destroyed by RISC. So this is one source of RNA for RNA interference. Now for second source, in your book they mentioned there are transposons in organism. What are transposons? Transposons are some mobile DNA segments, jumping genes. Sometimes these transposons, they can change their position. They can move from one chromosome to another chromosome. They can create mutation. So they are also harmful. Transposons are also harmful. Now sometimes these transposons, how they multiply, they produce double-stranded RNA. So this is a transposon. It's a transposon, it produces double-stranded RNA. This double-stranded RNA may be converted by dicer into small segment. So this is a small segment, it is produced from this RNA. This RNA is produced by transposon. Now this RNA is converted into small segment siRNA. This siRNA is formed with the help of dicer. Okay, dicer cut this and form small segment. Now this, from this small segment, one strand may bind to this RISC. One strand may bind to RISC. Now RISC is activated. If RISC is activated, then in future, if this transposon is trying to multiply inside the body, it produces RNA, and that RNA is destroyed by RISC. Okay, not always, it may destroy that. So this is second. Okay, this is second source for activation of the RISC. Now topic here is pest-resistant plant. It is tobacco. So they are talking about tobacco plant. In tobacco plant, nematode is common pest, and this nematode infection can be prevented by RNAi. So we will study that, how it prevent infection of the plant. Now scientists, what they have done in scientists, they isolated nematode specific gene. Scientists, they isolated some genes from nematode. Those genes are transferred into plant cell. This is a tobacco plant. Okay, this is a tobacco plant. This is nucleus of the tobacco plant. Into nucleus, scientists, they have transferred nematode specific gene. Okay, nematode specific gene. Now these nematode specific gene, they produce two types of RNA, sense and antisense RNA. Sense, antisense, they are complementary, so they will pair and form double-stranded RNA. Now when nematode feed on tobacco plant, this RNA may enter into nematode body. Now suppose this double-stranded RNA is inside nematode body. Now inside nematode, this inactive risk is already present. But now this double-stranded RNA, one strand of this double-stranded RNA, it bind to risk. Now this risk is activated. If this risk is activated, and now this risk, suppose this is mRNA of the nematode, now this activated risk, if it comes in contact with the mRNA of the nematode, it will destroy mRNA of the nematode. Now when mRNA of the nematode is destroyed, it will not produce protein. So this nematode, it will not survive. A nematode cannot survive because proteins, essential protein which is required by nematode, they are not synthesized. Why? RNA of the nematode is destroyed by this activated risk. Sorry. Now this activated risk is activated by which RNA? It is activated with the help of this double-stranded RNA which is coming from plant. Okay. How these are produced in plant? In plant, these RNA are produced by artificially introduced gene. So in your book, they are saying artificially genes are introduced in nematode plant. Okay. These are NCERT points. So they are saying nematode Meloidogyne incognita infect root of the plant. This is one point, and it is protected from this nematode. This tobacco plant is protected from nematode by RNA interference. It is a novel strategy. They are saying RNA interference is a natural cellular defense mechanism present in all eukaryotic cell. In RNA interference, it works by silencing specific mRNA using a complementary double-stranded RNA. Now double-stranded RNA bind to target mRNA and prevent its translation. As a result, gene does not produce protein. Now they mentioned there what is source of double-stranded complementary RNA. You can recall we studied one source may be viral RNA genome and second may be RNA produced by transposon. But these are not effective methods to control nematode in tobacco plant. Now scientists, they isolated nematode specific gene, and these are introduced into tobacco plant by using this bacterium called agrobacterium. Okay, this bacteria, agrobacterium, it is used as vector. Now they are saying inserted DNA produce both sense and antisense RNA. So here scientists introduced nematode specific gene. This nematode specific gene produce two types of RNA, sense, antisense, both will fuse. And now if nematode feed on this sense and antisense, this RNA enter into nematode. Inside nematode, it can activate a risk. So in your book, they mentioned double-stranded RNA initiate RNA interference. Double-stranded complementary RNA initiate RNA interference. So this double-stranded RNA, it enter into nematode. Inside nematode, one strand of this bind to risk. Now this risk is activated. If it is activated, it will start destroying nematode mRNA. If nematode mRNA is destroyed, it will not produce protein. If it is not producing protein, nematode will not survive. So you can prevent multiplication of nematode in tobacco plant. Okay, inside tobacco plant, we have introduced gene. This gene produce sense and antisense RNA. They are complementary. They bind and form double-stranded RNA. This double-stranded RNA activate the RNA interference mechanism. Okay, as a result, double-stranded RNA silence specific mRNA of the nematode, and nematode gene cannot produce its protein. Parasite cannot survive inside the plant. Okay, so finally, this nematode will not multiply. Now you can see this NCERT paragraph. They mentioned RNA interference takes place in all eukaryotic organism as a method of cellular defense. This involves silencing of specific mRNA due to complementary double-stranded RNA. What is meaning of this? Suppose you can see this is mRNA. We want to silence this mRNA. This mRNA is silenced with the help of RISC. RISC is activated by this double-stranded RNA. So this double-stranded RNA, source of this double-stranded RNA which activate RISC may be virus RNA virus, or source of this RNA may be transposon. Transposons are mobile DNA segments in eukaryotes. They produce RNA, and this RNA here can participate in RNA interference, or it can activate RISC. If it is activated, then it can destroy this type of RNA. Now you can focus on this paragraph. Here they mentioned this method involves silencing of the specific mRNA due to complementary double-stranded RNA molecule that bind to and prevent the translation of the mRNA silencing. Source of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic elements transposon. I told you transposon synthesize RNA and it can participate in RNAi. So they are talking about RNA intermediate which is produced by transposons. Now in next paragraph they are saying using agrobacterium vector, nematode specific genes are introduced into host plant. So nematode specific genes are introduced here. These nematode specific genes, they produce sense and antisense RNA. Now sense antisense RNA, they form double-stranded RNA. This double-stranded RNA is absorbed by nematode. Nematode can absorb this from external medium. And this double-stranded RNA, it initiated RNA interference. RNA interference how? Double-stranded RNA is present here. Now one strand of this bind to RISC. What is RISC? Protein complex plus RNA. So when this RISC or RISC is activated, this activated RISC, it will start destroying mRNA of the nematode. Okay. So here they mentioned two RNA being complementary. These are two RNA, they are complementary. They form double-stranded RNA. This is double-stranded RNA. This initiate RNAi. How it enter into nematode? In nematode, it activate RISC, and then RISC will destroy RNA. So they are saying it initiate RNAi and silence this specific mRNA. Silencing specific mRNA means destroying this RNA. So they can destroy this RNA. Now parasite will not survive. Why? Because RNA is required to produce protein. If protein is not formed, this nematode will not survive. Okay. So this is a method. It is called novel strategy. It is used here to control nematode pathogen in not pathogen but parasite in tobacco plant. Thank you.